I'm delighted to be here today. What I was asked to do is give a short overview of gene therapy. And I thought I'd do this for neurological disorders and then highlight some of the recent developments in leukodystrophies as I go along. So I hope I can make this short so I have enough time for questions. Here are my disclosures. So I will talk about, in general, approaches to gene therapy, highlight some of the challenges around potential toxicities, and as well as opportunities for leukodystrophies, and hopefully address some of your questions along the way. So the field of gene therapy has made tremendous progress, particularly in the past 10 years, but it's been a long time in the making, since the 1960s and 70s, when first viral vectors were developed, in particular what we call adeno-associated virus, AAVs, as well as first retroviruses that were applied to different diseases, initially to cystic fibrosis, and then also to immune deficiency syndromes. And then really over the past 10 years, there's been a whole accelerated development in the field of neurology and neurodegenerative diseases, with first human leukodystrophy starting to be treated as early as 2009. And we saw the past few years progress and approvals for spinal muscular atrophy, as well as retinal diseases. And I will highlight some of this. So when we talk about approaches to gene therapy, we're talking today mainly about in vivo AAV-mediated gene therapy, ex vivo lentiviral gene therapy, and antisense oligonucleotides. So what does that mean? And I will briefly highlight here the different kinds of viral vectors that are used for brain and spinal cord. The adeno-associated viruses are small DNA viruses that can carry smaller transgenes, and their effectiveness depends on the different type of AAV that's used. We call that serotype, and they have a depending on the serotype, they have a predisposition to go and target different parts of the nervous system, either brain or spinal cord, and different cell types. That's different than lentiviral vectors that are larger RNA viruses, and they're particularly good in integrating into the genome of the host, and they are good at delivering to bone marrow and blood cells in particular. So these two different kinds of viral vectors are used on the one hand for in vivo delivery, which is a direct delivery, often directly to the target organ or the compartment where this gene is needed. On the other hand, lentiviral vectors often are used to transfect bone marrow cells. These are cells that are taken from the patient, often then in a dish, transfected with a lentiviral vector, delivering a healthy copy of the gene, then delivered back. So that's why it's called in vivo, direct delivery, and ex vivo, where you're actually outside of the body performing a cell-based delivery. In vivo delivery can be directly into the brain. It can go into the spinal fluid space that you access when you get a lumbar puncture, but it can also go directly into the bloodstream intravenously. And this has been applied to different neurological disorders, and it's good to understand where the target gene is needed. On the other hand, I want to just highlight briefly the path that AAVs take inside a cell. So once you've delivered into the compartment, this AAV usually internalizes into the cell. It then has to travel through something we call cytoplasma and lysosome. And then it is usually deposited into the nucleus, where this virus can then, depending on its nature, whether it's a single strand or something we call self-complementary, it forms its own circular episome, and it can then continue to deliver the missing transgene. So very exciting. And this really led to a big change in the field of spinal muscular atrophy that for decades people thought was not amenable to treatment, and a very devastating disease that really in early infancy and childhood led to devastation. A lot of these babies unable to sit up, unable to stand, and they suffered from a spinal cord disease affecting the lower motor neuron. Now, this disease in particular suffers from a lack of SMN1 gene, and turns out you can only survive if you have at least one copy of SMN2, and that will be important to explain to you briefly how antisense oligos work. But first pass, it seems clear that one approach at least is to replace SMN1, and that is exactly what happened with the AAV approach that now a lot of companies are building on for other disorders. So SMN1 replacement and gene therapy really came out of the insight that AAV9 is a very good vector to target the nervous system, and particularly motor neurons in the spinal cord when injected into the spinal fluid, into the CSF, or intravenously. And as I mentioned before, the AAV9 then can sit in those cells and produce ongoing transgene to rescue those target cells in spinal cord. And this really led to a transformative experience in the field of spinal muscular atrophy. As I mentioned before, a lot of these kids unable to even sit up, and when one gave this therapy at low dose, there was some improvement, but when you really started to see high dose treatment, that's when things really took off. And for the first time, it was seen that these children could sit and walk and run and do things that had never been seen before. So this is obviously generating a lot of excitement, and we're about to also launch an intravenous AAV9 gene therapy for Canavan disease, building on the wonderful work of Guangping Gao at UMass. And this is here treating, again, children early in the stage of the disease, within the first 2 years of their life, and trying really to get ahead of the disease that often starts very early and affects not only white matter, but deep basal ganglia structures. And what we want to see is if after delivery, can this be done safely, and can we lower what is the culprit in this disease, the N-acetyl aspartate that is so high in this disorder, and can we lower that successfully? Another trial that is already underway at our center is an AAV-GM2 delivery to brain for Tay Sachs and Sandhoff disease. That is another devastating disorder of infancy, where we are now seeing children come to us from all over the world. This is a collaboration with UMass Worcester, where we are delivering not only into the intrathecal spinal cord space, CSF space, but also directly to brain. And this is done by a very skilled neurosurgeon, Dr. Katal Tepe, and you can see in both these cases, right after the delivery, there's a little bit of edema here in the thalamus, but that then disappears over the coming weeks and months. Another trial that's in planning stages is using an AAV9 is to the spinal cord for adrenomyeloneuropathy. This has been shown in my lab to work very well using an osmotic pump that in mice is inserted under the skin and can deliver into the lumbar space of the spinal cord. And we think this will be helpful for adult patients with adrenoleukodystrophy, patients known to have adrenomyeloneuropathy. And we know that we can target the right cell types in spinal cord and also lower very long chain fatty acids. And given some of the successes seen in other spinal cord diseases, we are quite hopeful. So shifting gene therapy, I told you this uses a different virus, the so-called lentivirus, and this lentivirus has a particular predisposition to target bone marrow cells, in particular hematopoietic stem cells. And this seems to work for brain diseases. And we think the reason that that works is that these hematopoietic stem cells go from bone marrow to blood and from blood to brain. And there they can renew a particular compartment of cells known as microglia. Whether it's just through that effect or through others mechanisms is not quite clear. But the exciting thing here is that really, because these are stem cells you are transfecting, you have a lifelong source of therapeutic protein produced out of the bone marrow. And just to highlight that we did this for adrenoleukodystrophy, as many people might know, particularly for the childhood cerebral form of the disease that affects the brain and really causes such devastation to boys who were initially developing normally. Why does this work? We think this works because we've seen in bone marrow transplantations that survival can be dramatically improved using this approach. And we think that these are the many microglia that are dying along the lesion edge are successfully rescued through these newly corrected cells. So we reported a few years ago a first success in 17 boys with childhood cerebral ALD who had evidence of contrast enhancement and at the time did not have a matched sibling donor for bone marrow transplantation. And we were able to show that we could improve the contrast enhancement, halt the lesion. And so far, we've not seen any adverse events of insertional oncogenesis. We see the same kinds of side effects that are seen in the conventional transplantation that pass after the critical period. How is this done? Usually the boys, when they have childhood cerebral ALD, we see them, we harvest their bone marrow cells after we mobilize them, usually with a compound such as GCSF. We take those cells out of the body, we select a certain type of stem cell, the CD34 positive cells, we transfect them in the dish with a lentiviral vector, as I mentioned before. This gives them a healthy copy of the faulty gene, and then we deliver those cells back after a brief amount of chemotherapy. And then we follow them for a 2-year period. So everything very similar to conventional bone marrow transplant, with the exception that these are cells from the boys themselves. So far, this has shown good results in getting not only the viral vector into the blood, but also expressing now healthy ABCD1 protein, or ALD protein as we call it. And this has now been done in more than 50 boys across the world. And recently EMA not just recommended approval, but now is given the go-ahead for marketing in Europe, and filing of this drug in the US is about to happen with FDA. And just to briefly highlight that contrast enhancement in most cases goes away at 6 months. We still see a little bit of stuttering over time, but the effect of this is usually that the lesion stops growing. So this approach has been not only helpful for adrenoleukodystrophy, but also for metachromatic leukodystrophy, where there is already approval in Europe. And we hope that over time, there'll be also further brain disorders that are added to this list. Turning back to SMA, I mentioned before that there's a SMN1 copy missing. What I didn't say is that depending on the number of SMN2 copies, which is a protein that has very close similarity to SM1, the children have a variability in severity. So those kids who have only 1 to 2 copies of SMN2, they are most severely affected and never sit. Those that have 2 to 3 copies, they sit, but they never stand. And those with 4 or more copies, they stand and walk. So the idea arose, is there a way not just to give back SMN1, but somehow improve the copy number of SMN2? And it turns out that SMN2 is usually truncated and degraded, but there is some residual SMN2 that is produced. And if you can prevent this type of alternate splicing that occurs and degrades the SMN2, then maybe there's a way of boosting the amount of protein. And this is exactly what happens through this antisense oligonucleotide, where splice switching can occur. Let me briefly explain what antisense oligonucleotides are. These are chemically modified single-strand DNAs. Different than viral vectors, these are very short pieces of DNA molecules that target RNA. So whatever is encoded by DNA into RNA is what this interferes with. So it is taken up and enters nucleus, and there interferes with the RNA. And in the case of SMN2, it helps lead to this splice switching so that SMN2 is not degraded. And this led to a very successful trial in spinal muscular atrophy, where they were able to show in the NURTURE trial, after repeated treatment intrathecally with this antisense oligo, similar to what was seen after gene therapy, an improvement in function, a gaining of milestones that had never been seen before. So are we seeing this now? Just to mention here, this has already been approved by FDA in 2016. So in the leukodystrophy field, we're seeing similar approaches move forward for diseases like Alexander disease. Alvin Messing showed very nicely that he could use these antisense oligonucleotides to deliver to mice affected by Alexander's disease, and there not just reduce the toxic GFAP mutant protein, but also prevent the pathology that was arising in these mice, the so-called Rosenthal fibers. And we're particularly excited that now there's a multi-center trial that has just opened June 1st and is treating really a wide range of patients with Alexander's disease, ranging from 2 years of age up to 60. And we're delighted to be part of this effort by Ionis. So just to mention in closing that there are potential toxicities of all of these three approaches. On the one hand, in vivo AAV gene therapy can lead to inflammatory reactions. You can see a rise in certain liver enzymes, which is why these children are often placed on oral prednisone, so that the immune suppression avoids this type of inflammatory reaction in the liver. In animals, there has been oncogenesis, meaning tumor formation seen, but so far we have not seen this in any human studies. So it's something we're very cautious about, and we're watching whether there are any so-called clones that arise, but so far this has not been a concern. As I mentioned before, liver pathology can occur and is something that particularly at high doses of intravenously delivered AAV needs to be handled with much caution. A lot of trials using immune suppression, immune modulation to avoid this. And another part of the nervous system that we've also seen inflammation in is a part next to the spinal cord called the dorsal root ganglia. These are also places where we think we can avoid inflammation by giving steroids before the gene therapy is delivered. On the other hand, in ex vivo gene therapy, you can see the kinds of events that occur also in a conventional bone marrow transplantation: nausea, vomiting, infections, fever. Early on in ex vivo gene therapy performed in severe combined immune deficiency, there were some cases of leukemia that occurred, but with all the modern day lentiviruses, this has not been seen. And just to highlight again that we're monitoring these kids closely to make sure that there are no single clones that arise in blood or out of those bone marrow cells. What happens with antisense oligonucleotides? Obviously, because you're giving a lumbar puncture, there can be a post-LP headache. Sometimes there are challenges because there are deformities of the spine and the intrathecal space, the space around which the CSF is, cannot be delivered into well. Some patients have suffered from some kidney problems and low platelets, but overall this has been well tolerated and something that trials have accumulated much experience in hundreds of patients. So just to, in closing, mention that I think we're now treating diseases that we previously thought were untreatable. I've showed you at least 3 different approaches that are all correcting the faulty gene, but the right approach has to be found depending on the underlying neurobiology, what cell types are affected, what part of the brain or spinal cord are affected. I didn't talk much about things like promoters, but you have to understand how much expression you want and what cell types, and that can often be guided by the use of certain promoters that are being used in the construct. As I mentioned, the timing of the treatment is key. Many of you know that if you treat too late, often not only is the treatment ineffective, but sometimes harm can be done. And that's why we have to be very careful about understanding balance of safety and efficacy using clinical trial criteria. And I think Dr. Bunkowski will now be talking about that. So I think overall what we've seen is really a paradigm shift in the past 10 years and many more trials coming our way. When I first opened my clinic 15 years ago, most boys came to me in wheelchairs and really at death's door. But I think today I see most of the boys in my clinic who are sending me pictures, like this boy on the right, reporting from soccer camp and leading active lives. So it's something I celebrate, but I also do caution that one has to do things one step at a time with safety first. So I think that was my last slide. I'm glad, Anna, to take any questions and